Okay, this is a short video trying to describe the science behind my biotechnology project for my AP biology students. This project is going to revolve around a little circular piece of DNA called a plasmid. And as you can see with this diagram, what we can do with a plasmid is we can cut it open with the protein, and then we can insert foreign DNA into it, so DNA from another source, and then we can make a recombinant plasmid, meaning this particular piece of DNA has two sources of DNA, right? One is from the foreign source, one is from the bacteria, which that's where plasmids come from. Plasmids are responsible for antibiotic resistance in bacteria, which is one of the um, the concepts that we're going to be focusing on in this project. Pretty much, how do bacteria acquire antibiotic resistance? Okay. Another kind of application to plasmids and this recombinant DNA technology is insulin production. Right. When we have somebody who's diabetic and they are insulin dependent, that insulin does not come from little human factories. They come from little bacterial factories. We actually make a plasmid that has human insulin gene, and then we make the bacteria produce the protein, the insulin, and then we harvest it. So we're going to do something similar. We're going to be harvesting, um, not so much harvesting the protein itself, but I'm actually wanting to harvest the plasmid itself. We are currently doing a lot of biotechnology in AP biology, but it's expensive. We're talking probably hundred to thousands of dollars an ounce for some of these reagents. This project will allow my students to engineer their own plasmids as well as provide a stock. So I'll have an actual back stock of these materials that I will not require to purchase them every year because I'll have a stock of them. So the process of harvesting this plasmid from the bacteria, we're going to do something called a mini prep. So pretty much I take the bacteria, I'm going to break them open, and then I'm going to harvest the plasmid out of them. And actually, I won't. My students will be doing this. This is a lab skill that is present in many biotechnology labs, so they're going to get the exposure of actually doing some real life biotechnology and not just stuff that happens to be from a kit. In fact, another way to look at this is they're making their own kit here, and they're essentially making the kit for the following year's class. So I'm not going to go in too far into the process of the mini prep, but if you have any questions, you're more than welcome to ask me. And then the last thing we're going to do is once we have all the DNA and because all the plasmid is harvested, we'll analyze it with gel electrophoresis, right? This is a gel. Many of you have seen something like this on CSI, or you've seen something like this on NCIS, where they're looking at, crime scene investigations and they're analyzing DNA. This is going to be no different than what we do in class. So if you are interested in donating, just go ahead and go to Donors Choose and search my name, Rapasura. Okay, and any and all contributions will be welcomed.